Hello everyone. In this class, we'll learn about action potential. Action potential can be defined as a sequence of changes in the membrane potential of an excitable cell due to opening and closure of different ion channels after the application of a threshold stimulus. This is the graphical representation of the change of the membrane potential when a nerve is stimulated. The membrane potential is going to change from minus 70 to plus 30 millivolts. So when the stimulus is applied on the nerve, initially there will be entry of the sodium channels, which causes the depolarization, and at this point opening of the potassium channels, which is going to lead to the potassium efflux, and there will be loss of potassium ion that is going to lead to a repolarization. And we have different uh, other stages also, like stimulus artifacts, firing level, overshoot, after depolarization, and after hyperpolarization in an action potential. We're going to see them one by one. As I said, there are different phases, that is resting membrane potential stage. So here the inside the nerve, the potential is negative, and outside the nerve it is positive. So that's why the resting membrane potential is always negative inside the cell compared to outside the cell. The membrane is maintained in polarized state by due to the distribution of ions across the cell membrane, and some ions diffuses out of the cell along its concentration gradient, while non-diffusible ions, that is proteins, stay inside the cell. If you compare inside the cell, there are some negative ions, and because of which there will be uh, unequal distribution of the ions which is going to occur. And if you know, and we know that potassium concentration is high inside, and this tries to move out of the cell, and at the same time sodium concentration is high outside, which is going to try to move inside the cell. And the permeability nature for potassium is high, so sodium cannot compensate for that. So uh, the the negative inside will be more because of the potassium uh, efflux. As we said, sodium influx does not compensate for potassium efflux because the membrane at rest is less permeable to sodium than potassium. And also sodium potassium pump causes continuous pumping of 3 sodium to outside for each 2 plus potassium ions pumped inside. This also creates additional degree of negativity, and that is the basis for resting membrane potential. And stimulus artifact is when stimulus is applied, there is a brief irregular deflection of baseline due to the current leakage from stimulating electrode to the recording electrode, and the latent period is going to corresponds to the time it takes to travel along the axon from the site of stimulation to the recording electrode. So this diagram showing the resting membrane potential, it is always maintained negative inside, positive outside. It is mainly because of the potassium efflux which occurs, and also there is because to compensate, the sodium is going to try to move in, but the potassium overrides that activity because it has a more permeability for the cell membrane. So the the inside of the cell, that is why it is maintained, it becomes more negative. And also sodium potassium pump, which tries to uh, pump 3 sodium uh, out of 2 potassium, so that is also is responsible for some part of resting membrane potential. Depolarization, when the membrane potential reaches the voltage between minus 70 to minus 50, there occurs sudden conformational change in the activation gate of sodium channels, flipping into open state. That is, these are voltage gated sodium channels and has a two separate gates, activation and inactivation gate. Uh, as activated state sodium ions, they are going to move in. The tracing rises rapidly till plus 35 millivolts. Now inside the nerve cell becomes positive and outside the nerve it is negative. You can see, so when the voltage gated sodium channel, they are going to activate, so it is going to cause activation gate to open, so that is why sodium is going to move inside. Okay, and at 30 mmFHE, the inactivation gate is going to close, and that is going to limit the entry of sodium, and it is not going to open until the resting membrane potential reaches back to uh, resting membrane potential. And when some sodium enters inside the cell, okay, and it is going to lead to the uh, threshold level, that is at threshold level it is going to open up the adjacent voltage gated sodium channels, and so this is going to cause a tremendous entry of sodium ions inside the cell, and that is going to lead to the massive change in the membrane potential. Okay, and the stimulation of adjacent voltage gated channels, I mean, is called as Hodgkin's cycle. That is, the change in the polarity is going to stimulate the adjacent sodium channels, and the causes the entry of massive amount of sodium ions is called as Hodgkin cycle. Repolarization. After sodium channel has remained open for 10,000 of a second, it suddenly closes. At this point, the membrane potential begins to recover back to resting membrane potential. Repolarization starts with potassium efflux due to the opening of voltage gated potassium channels. A very important characteristic of sodium channel inactivation process is that inactivated gate will not reopen until the membrane potential returns to resting membrane potential. So this is the ionic basis of repolarization. At minus 35, the potassium channel is going to open, and this is going to lead to the potassium efflux, and so this potassium loss continues till the hyperpolarization state. After depolarization, at the termination of spike potential, potassium conduction is slowed down, and thus few milliseconds are delayed in restoring membrane potential. So this last phase of slow potassium efflux is called as after depolarization. And after hyperpolarization is where resting membrane potential is achieved by the active sodium potassium pump mechanism, which transport 3 sodium out and 2 potassium in. Okay. So after hyperpolarization occurs because of the delayed closure of the potassium channels, which causes the more negativity inside the cell because of the delayed potassium efflux, and by uh, putting out some sodium outside the cell, uh, it is going to bring back the resting membrane potential back to the normal. Okay. So that is done by sodium potassium pump. Okay, so this is the ionic basis of after depolarization. It mainly occurs because of the increased potassium levels outside. So that is why the potassium efflux is delayed. So that is why the repolarization process is delayed in this part. And hyperpolarization occurs because of the continuous or delayed closure of the potassium channels, and the resting membrane potential goes beyond the resting membrane potential, and then it is recovered by the sodium potassium pump. Properties of action potential. Threshold stimulus. It is the minimum intensity of stimulating current that after acting for a given duration will just produce an action potential. The relationship between strength and duration of stimulus has been studied by varying the duration of stimulus and finding out the threshold strength of each duration. rheobase and chronaxy. The weakest current strength which can excite a tissue is, if allowed to flow through it and uh, for an adequate time, is called as rheobase. The time for which it is applied is called as utilization time. The length of the time for which the current of twice rheobase intensity must be applied to produce a response is called as chronaxy. Okay, so this diagram shows the relation between the duration which is required for the excitation and the strength of the stimuli. Okay, and we need the lowest strength of stimuli that is required for the excitation of the tissue is called as rheobase, and the time required for this is called as utilization time. And if you are using the rheobase, that is the twice the time required for excitation of the tissue, and the time required when you use 2 rheobase, it is called as chronaxy, and it is the parameter is used to assess the excitability of the any tissue. It has all or none response as well. The action potential is an all or none response to stimuli. That is, if stimulus is subthreshold, not adequate, no action potential is produced. Once threshold is reached, a full-fledged action potential is produced. Further increase in the intensity of stimulus produces no increment in the height of action potential, provided the other experimental conditions remains same. Accommodation. It is an excitable cell is applied with a constant strength of current. The site of membrane under stimulation fails to produce action potential, that is, the membrane adapts to the applied stimulus. This is due to the slower opening and delayed closing of the voltage gated potassium channels. Conductivity. Uh, the action potential developed at one location on the excitable cell acts as a stimulus for the production of action potential in the adjacent region of the membrane. The excitation impulse is conducted along the cell membrane as a wave of depolarization. This is another property of the action potential. Refractory period. If two successive stimuli for more than the threshold intensity are applied to an excitable cell, it is found that for some time after the first stimulus, the cell becomes refractory. There are two types of refractory period, that is absolute refractory period and relative refractory period. Absolute refractory period begins from the time of the firing level is reached till the repolarization is approximately 1/3 complete. During this period, no strong stimulus can initiate fresh impulse. And the relative refractory period begins at the end of absolute refractory period to the start of after depolarization. And during this stimuli stronger than the normal stimulus can cause excitation. So this is diagram showing the the refractory period, that is the absolute refractory period, and the relative refractory period. And the absolute refractory period is till the 1/3 of the repolarization, and the relative refractory period will be after the 1/3 of the repolarization, where during absolute refractory period the second stimulus will not produce any stimuli whatever the strength of the stimuli, but in relative refractory period the higher strength of stimuli sometimes it can evoke an action potential. Saltatory conductions. Depolarization in myelinated axons jump from one node of Ranvier to the next, and this jumping of depolarization from one node to another node is called as saltatory conduction. It is a rapid process, and myelinated axons conduct up to 50 times faster than the unmyelinated fibers. So this diagram showing the saltatory conduction when in the node of Ranvier, and this the fiber is myelinated. When action potential is produced in one node of Ranivir, the positive ions electrotonically conducted to the next node of Ranivir, where it will be able to produce threshold stimuli, and there will be opening of the voltage gated sodium channels, which is going to produce another action potential. So similarly, the process is repeated to the next node of Ranivir, which is going to hasten the conduction of the impulse. Monophasic action potential. In this, one microelectrode is placed inside the nerve fiber and other electrode on the outer surface. These electrodes are then connected to the cathode ray oscilloscope. Monophasic record has the following components: depolarization and repolarization. Biphasic action potential. Here, both the electrodes are placed on the outer surface of the nerve fiber and they are connected to cathode ray oscilloscope. When nerve is stimulated, a record of alternate reflection, one positive above the baseline and one negative below the baseline, is depicted. So this is a diagram showing monophasic action potential, and so this is on the normal nerve fiber, and this is on the mixed nerve fibers. Okay, in the simple nerve fiber which is not mixed, you see only one peak, whereas in a mixed nerve you are going to see the different peaks. Compound action potential. Compound action potential is the monophasic recording of action potential from a mixed nerve which contains different types of nerve fibers with varying diameter. Therefore, compound action potential represents an algebraic summation of all or none action potential of many axons. The causes of compound action potential are a mixed nerve fiber is made up of families of fibers with the varying speed of conduction. Therefore, when all the fibers are stimulated, the activity in fast conducting fibers arrive at the recording electrodes sooner than the activity in slower fibers. And the farther away from the stimulating electrodes the action potential is recorded, the greater is the separation between the fast and slow fibers. In general, group A fibers make a maximum contribution to compound action potential and group C to the least. So this is diagram showing the compound action potential. You can see the separation of the action potential of different diameter of nerve fibers. Okay, so we can see the A alpha fibers, their action potential reaches very fast, and followed by B, A beta fibers, gamma and delta fibers. So then it is followed by B fibers and C fibers. So this is in brief about action potential. Thank you.